So I'm most grateful for this kind and generous introduction, for the welcome, for the invitation to be here, to all of you for coming to participate in this event. I was reminded by the first of the remarks made by the prorector of the fact that experimental science actually began in Denmark with the work of Tycho Brahe. Experimental science started when Brahe made his measurements of the movements of the stars and planets with great precision, with increasing precision, and this dovetailed with the theoretical work of Johannes Kepler at the same time in Europe. And it was really through their combined efforts that the enterprise in which we are engaged today found its beginning and we hope will never end. I thought I should just comment briefly about the title, which may seem presumptuous. It's actually partly facetious or self-mocking. The reason is that at least the older members of the audience will recall a similar title of a treatise written by a man named Francis Fukuyama. It was entitled The End of History, published about 30 years ago. As we all know, history hasn't ended. Nevertheless, I intend something serious by the title as well, which I hope to convey in the course of this lecture. I'm going to tell you a brief scientific story from my own experience, and then I will be so presumptuous as try and draw from it a larger meaning and connect it with something which I believe has much wider significance, and I hope indeed to many of you. Something which we might discuss further in the period that follows the lecture, which I much look forward to. So the scientific story that I'll tell you begins with the genome. It is, as you will know, most significant for its permanent, its conserved, its static nature. It is through precise replication of the genome without ever making a mistake, that the information, genetic information, is transmitted from parent to offspring. This is the basis of heredity. The information in the genome, as has just been mentioned, is utilized through the process of transcription for the synthesis of RNA, and then translation by the ribosome for the generation of proteins, which are in turn responsible for all the form and function of a cell, indeed of every living cell. We used to think of this as essentially static in nature as well, that the structure of a cell was laid down, that its functional potential was established, and that it never changed. Remarkably, in the last couple of decades, it has emerged, the process is remarkably dynamic. Information from within the cell and also from outside is transduced through pathways leading to transcription, to regulate which genes are transcribed in what place at what time. The process is, as I say, remarkably dynamic. It reshapes the cell on a continual basis in response to environmental and internal information. It is a process which plays a central role in everything ranging from immunology and the response to invading organisms and disease, all the way to memory and function of the brain. The dynamic nature, the information processing takes place in the nucleus, which serves as a kind of central switchboard. The information processing, which leads to the control of transcription, is referred to as transcriptional or gene regulation. And that is what I will primarily speak to you about today. Now, gene regulation begins with the genome itself, and indeed, as has been mentioned, especially with the structure and organization of the genome. When I was about the same age as most of the young people in the audience, I proposed on the basis of X-ray and biochemical studies the wrapping of DNA around a set of 8 histone molecules and a particle that has come to be known as the nucleosome. The nucleosome serves as the fundamental or organizing principle, the basis for packaging all DNA in eukaryote genomes, but it serves an even larger purpose. Some years later, we discovered the nucleosome interferes with many DNA transactions, so the wrapping of DNA around histones in the nucleosome prevents the initiation of transcription, an observation we first made from studies of transcription in a cell-free system, so in vitro. But soon after, it was shown the nucleosome subserves a similar function for the control of transcription in vivo. The nucleosome then serves as a general gene repressor. It prevents expression of all the many thousands of genes in eukaryotes, except those whose transcription is brought about by specific positive regulatory mechanisms. Now, we and others have investigated those mechanisms, and much progress has been made over the past 30 years. I won't talk to you about that aspect today, but rather pass immediately from what happens when the nucleosome is removed, the DNA is unraveled and made available for interaction with the transcription machinery. And in our case, we focus especially on the RNA polymerase II transcription machinery for a reason that is well known, I'm sure, to many of you. RNA polymerase II is responsible for all synthesis of the messenger RNA that codes for proteins in eukaryotic cells. And as the first step in the pathway of gene expression, polymerase II transcription is a focal point of cellular regulation. Most of the signal transduction pathways that I alluded to in a previous slide culminate in the control of RNA polymerase II transcription. Indeed, it is the intricate regulation of polymerase II transcription that underlies cell differentiation and development. The machinery for transcription extends beyond the RNA polymerase itself. There are many additional components. We and others discovered what are called general transcription factors that go by the letter names B, D, E, F, and H. The polymerase alone is incapable of unwinding DNA and the synthesis of RNA, as I will show you in a snapshot at atomic resolution in a moment. But in the absence of general factors, the polymerase is unable to start the process. It can't find a gene. It can't begin transcription. The most complex of the general factors rivals in size and complexity the RNA polymerase, and it plays an essential role not only in transcription, but in 2 other seemingly unrelated but equally important cellular processes: DNA repair and control of the cell cycle. In respect to DNA repair, it may astonish you to know that the many possible insults to our DNA result in on the order of trillions or even thousands of trillions of DNA lesions in every individual every day. And the reason why these do not have profoundly deleterious effects is because of repair by enzymes that correct almost all such mutations. Principal among these is the enzyme general transcription factor H. Evidence for this comes from humans mutant in this factor, which is the cause of a disease, as I will explain in a moment. First, I must clarify there are 2 kinds of repair process, one called base excision repair, which involves recognition of damage, then excision of the damage to leave a gap in DNA, which is finally closed. The other more prominent form of repair, nucleotide excision repair, also involves recognition of damage, excision of a larger segment of DNA that includes the damaged region, and then finally, once more, filling the gap to restore the DNA to its original undamaged condition. Now, as I mentioned, there are mutations in humans that interfere with nucleotide excision repair. They are alterations of the general factor H and the disease that is a consequence called xeroderma pigmentosum. It results from extreme sensitivity to UV light that causes DNA damage, and that leads in turn to a high risk of skin cancer. And the only remedy at the moment is protection by the use of special UV opaque apparatus. Now, there are additional lesions that affect proteins such as so-called CSA and B that couple transcription to repair. They lead to a disease called Cockayne syndrome, also characterized by sensitivity to UV light, but in addition which leads to neurological abnormalities and progeria, which is premature aging. Now, the general factors, as I have mentioned, interact with RNA polymerase to enable the initiation of transcription. Gene regulation is due to molecules that are either positive acting, so-called gene activator proteins, or negative acting, gene repressor proteins. These enter the nucleus of a cell. They contact recognition sites called enhancers in front of genes and impinge upon the transcription machinery to turn a gene on in the case of an activator, or to prevent its transcription in the case of a repressor protein. My colleagues at Stanford discovered some years ago there is an additional component required for communication between an activator protein and the general transcription machinery. Because it subserves an intermediary role, it goes by the name mediator. Especially my colleagues, Stefan Bjorklund, now a professor in Umeå, and Young-Jun Kim, presently in Korea, reported in 1994, mediator is made of dozens of proteins. It has a total mass greater than 1 million molecular weight. Over the years as it has emerged, mediator receives and processes all transcriptional regulatory information. Following information processing, delivers an appropriate signal to the RNA polymerase. And this occurs in the entire range of eukaryote organisms, from single-cell creatures like yeast and other fungi, to the most complex like humans and higher plants. Aberrations of regulation by mediator underlie a wide variety of human disease, including infectious disease, and, as this slide indicates, cancer. So here you see diagrammatically the 31 protein human mediator, and virtually every component is implicated in a variety of malignancies. It is interesting that in very many cases, for example, breast cancer, bladder cancer, the aberration of mediator is an overexpression, a superabundance of one of the components of the mediator complex, not a mutation of a component of the complex. And I can suggest, if someone would like in discussion later on, why this kind of aberration, simply a superabundance of a subunit of the mediator complex, should have such a high association with cancer. So in the case of both breast and prostate and bladder cancer, a particular subunit of mediator is overexpressed in more than 50% of all individuals suffering from the disease. To summarize then, the machinery for transcription, polymerase, the general factors, and mediator, constitute nearly 60 individual proteins that assemble in an object of greater than 3 million molecular weight, so larger than a ribosome particle, at every gene prior to every round of its transcription. Francis Crick, who you heard mentioned was one of my teachers, was supposed to have said, if you wish to understand functions, such as we do of mediator, study its structure. As I often say, when I made that remark to my father, he said, if you wish to understand structure, study function. Well, regardless, my colleagues and I have pursued the structure of the transcription machinery, and I'm going to tell you one of the findings which serves as a prelude for the connection to disease that I will explain later on. That one finding comes from investigation of the structure of the RNA polymerase itself. And this we did over a period of years by isolation of the enzyme in the formation of large single crystals. And these in turn could be solved by X-ray diffraction because the protein was at least 10 times larger than any previously studied. We required a source of X-rays much more intense than those previously utilized for this purpose. And they became available to us in the year 2000 from photon physicists at the Stanford Synchrotron Radiation Laboratory. The Stanford Synchrotron lies at the end of a 1-mile-long linear accelerator that you can see here in the distance in the background of the diagram. The electrons accelerated that 1 mile of length, then circulate around a ring, and in consequence of that motion, they emanate X-rays, and these are of especially high intensity. It took us 10 years to purify and learn to grow suitable crystals of the enzyme. It took another 10 years, 10,000 liters of cell culture from which the enzyme was derived, and one graduate student went into the solution of the problem. What you see here is my longest serving associate, Dave Bushnell, lowering a graduate student, Ken Westover, into the 100-liter fermenter to repair a valve at the bottom. Ken was a medical student and graduate student at Stanford, a so-called MSTP student. He went on to complete the requirements for both graduate and medical degrees, then did clinical study, so was a resident and a research fellow at one of the Harvard hospitals in Boston, and is now a clinician scientist at the University of Texas Southwestern in Dallas, Texas. He's a very fine scientist. Ken was at the time also a gymnast, and so slender and could fit into the fermenter to repair the valve at the bottom. And I often say, I wonder where we'll ever find another student of his caliber. So anyhow, we solved the structure of this enzyme by X-ray diffraction. We located the positions of almost 30,000 carbon, nitrogen, and oxygen atoms to near atomic resolution, arriving at the picture that is shown in this slide. Here, each of the individual subunits in a different color is displayed in the form of what is called a ribbon diagram that indicates the path of the polypeptide chain of the particular protein. The one that is colored purple at the lower left represents the 5th largest subunit of this giant complex. It is a 25,000 molecular weight protein, so a typical sized protein, which gives you a sense of the size and the complexity of the entire object. I'll rotate the picture around to a view down the central channel that I will return to in a moment. The first thing you wonder when you look at this enormous complex, the RNA polymerase structure, where does the gene DNA enter for transcription? From what point does the product messenger RNA molecule exit? Well, you might imagine that the gene DNA enters through this capacious central channel that I called your attention to a moment ago, that leads to the point marked by this pink sphere, which symbolizes the location of a magnesium ion required for catalysis, and so that identifies the active center. We proved that the DNA enters at this point, and RNA from the location of the magnesium ion by solving the structure not only of the enzyme alone, as you see here, but also the enzyme in the form of an actively transcribing complex with the gene DNA present and the RNA in the act of synthesis. I'll show you that in a moment, but first a color code to the slide that follows. It had been known for many years that about 15 base pairs of the DNA double helix are unwound in the form of a so-called transcription bubble at the active center of any polymerase engaged in a process like transcription. It was furthermore known that some 8 to 9 base pairs of RNA-DNA hybrid are centered within the bubble, with the growing end of the RNA adjacent to the catalytic magnesium ion, 3 residues from the single strand